Hello to everyone to a short talk about June and its connections with Wnt and Notch. Unfortunately, Gerlinde, she couldn't make it to make this video, so I'm happy to give you this overview. Regarding the structure of this talk, I will first give you some background information about June, then I will move on to the hypotheses that we want to answer. Then we will come to results that we have gathered so far and what we still want to do to find answers to our hypotheses. And finally, a summary. Regarding June, there are a lot of different things to know about June. Just to give you some short facts about June. June is part of the AP1 transcription factor family. For example, there are June B and June D, so June is not alone. June also drives cancers and fibrosis, like IPF. June is also involved in a large variety of cellular processes, like proliferation and survival. And finally, last but not least, June also interacts with the immune system, like T cells, like macrophages. What are the questions that we want to answer? Two big chunks. The first one is that we want to see how June affects beta-catenin and Notch, and vice versa. How does beta-catenin affect June? And the second chunk of hypotheses is how do neutrophils affect desmoids? Does neutrophils part of the innate immune system not really well explored, but they are important, and we really would like to figure out how they contribute to desmoids. Do they drive desmoids, and through which cytokines? Now let's move to our results. In these results, we want to answer 5 different questions. The first one concerns how does activated June correlate with beta-catenin and Notch in desmoids. The two questions that will follow are in vitro questions, and the first one is to see how does June influence the desmoids in vitro, and the second one is how does June interact with beta-catenin. Then we will see how do neutrophils affect June and beta-catenin before checking in vivo. Is it possible when we combine June and Notch inhibition? Is it possible to slow down or even eliminate desmoid-like tumors in mice? Regarding the first question, how does activated June correlate with beta-catenin and Notch in human desmoids? What we saw in the past is that in these samples, June is expressed, and that a large portion, about a quarter of the genes that are commonly upregulated in desmoids, that they're also June targets. The problem with this antibody is that this antibody cannot differentiate between the unactivated dephosphorylated and the activated phosphorylated version of June. This is why we want to move this question forward and to see is phospho-June expressed in desmoids, and to which extent does it correlate with beta-catenin and Notch. We have stained 8 samples so far against beta-catenin and Notch. The phospho-June stain is the next, and then probably the sample size of 8 is too low, so we will need to add some further samples. The second question is how does June affect desmoids in vitro? One of the problems that we found in the past is that it was not easy to successfully establish these desmoid cultures in vitro. But fortunately now, we finally made it to generate June knockout and June overexpressing cultures, and we will analyze these cultures for cell proliferation in the near future. I can tell you from the observations under the microscope, there's a strong difference, but of course we want to measure the changes in proliferation objectively through an EDU assay, and we will stain some protein lysates, again, proliferation markers like phospho-AKT. Then, how does June interact with beta-catenin? Maybe you can remember this core-IP assay that we were also shown last year, and I can very well remember your criticism about this assay because this antibody against beta-catenin is very sticky. We have taken this criticism very seriously, and now in the next weeks or months, we will find time to confirm this assay. One possibility is to use a mass spec, and the second one is to use a proximity ligation assay. Additionally, now that we have these reliable June knockout and June knockout cultures and control cultures, we will use a reporter plasmid if June regulates beta-catenin activity, and we will also use beta-catenin G3 reporter plasmid to see if the June expression level, does it affect the physical distribution of beta-catenin. For example, just a hypothesis that when we induce June, it causes the beta-catenin to move into the cell nucleus. We will see this. Regarding the question how neutrophils affect June and beta-catenin, we at the beginning used the co-culture experiment. So we took blood, isolated the neutrophils, and then we had desmoids. And to one of the desmoids, we added the neutrophils, and the other one were just the desmoids. Kept this culture for 4 hours, and then we were running flow analysis. There was a slight effect on June, but a very strong effect on beta-catenin. And what we will do next is, of course, we need to confirm this experiment. So for another flow run and IF. And then even more interesting is to see which cytokine does cause this increase in beta-catenin. There are a large of different cytokines that are secreted by neutrophils. We will maybe pick 5 of them, and we'll see if these cytokines affect beta-catenin. And our last question, when we combine June and Notch inhibition, is it possible to slow down or eliminate the growth of a desmoid-like tumor in mice? And in these mice, we induce June locally. Compared to the spontaneous model, it's more reliable because there are more mice that develop these desmoids. And at the same time, we first induce the desmoids for 3 weeks, and then we will give the treatment for 8 weeks. We have one group without a treatment, one group with the AP1 inhibition, and one group with the combined Notch and AP1 inhibition. And then after 8 weeks, now we are week 5, the mice will reach the endpoint, and we will do an analysis for histology and also flow cytometry to see if there are changes in the immune infiltrate. So these are our results. Short summary, what we know so far, we know that June is expressed in human desmoids. What we don't know is really, is it the phosphorylated or dephosphorylated version? We know that about a quarter of the overexpressed genes in desmoids are targets of June. Now we have successfully generated our June overexpressing June knockout cultures that we will further use to run objective proliferation assays. We know that the core-IP assay shows a proximity between June and beta-catenin, but we will confirm this result. We have seen that neutrophils upregulate beta-catenin. We want to confirm this. In addition, we want to find out which cytokine is the underlying source. And we know that when we induce June locally, it causes desmoid-like tumors. And what we now want to know is, when we treat these mice with an inhibitor against June and Notch, does it eliminate or at least slow down the tumor? These are our results. We're really looking forward to your feedback and your suggestions. And yeah, I really hope to come to Philadelphia next year and taking part in the race. Yeah, thank you very much, and see you soon.